the way, I mentioned that the recordings are being made of these trust talks. Just to make sure everybody knows, all those trust talks that have been recorded are on the Trust YouTube channel. And currently there are 58 trust talks there. So a great resource for those of you that hear about a past talk that you want to watch. Well, with that, what I'm going to do is I'm going to share my screen, and I'm going to tell you a story this evening. And it begins with the whole idea about how salt marsh estuaries function, new answers to old questions. And to understand why I'm giving this program, I need to give you a little background. In 2018, there's a conversation between scientists at the DNR's Marine Science Institute and me. Basically, I went and asked them the question, is there a scientific summary of how estuaries function? The response was no. My idea was, why doesn't your scientific team do this? The director's response was, we are funded 100% by grants which focus on commercial seafood species. You asked the question, you should do this. So with that, then that got me going down the road to find the answer to the question that I asked. And that now 2 years later has led to this story. The story I'm going to tell you, is in the form of 5 chapters. The first is called the Fecal Coliform paradigm. The second is the Spartina paradigm. The third is the heyday of scientific research on salt marshes. Chapter 4 is in search of a new paradigm. And chapter 5 is the trust role in creating a new paradigm. So it's important to recognize that science starts with a paradigm. A paradigm is a proposed explanation, and essentially it's a story that scientists make up that shapes the questions scientists ask. Everyone that's listening has been involved and has used paradigms. Probably the best example, if somebody says, what's a healthy diet? Remember the days when everything was low cholesterol and they told a story about what was a healthy diet? Then came the Atkins diet. That was a paradigm because it told you what you should eat and why and how. Today, we've got a new paradigm that really focuses on gut flora, 21Me and all. And so if you recognize each of those is basically has some data and results in a story of what questions we should ask and how we do it. Importantly, a paradigm tells us how to look at the world. So the key thing here is that scientists, like all humans, are biased observers. And you have to understand biases in order to interpret what science finds. So with that, let's state about how things currently are used to evaluate the environmental health of Port Royal Sound. Of course, most of you know Port Royal Sound is the area where we're located. It's right at the southern tip of South Carolina. And what makes it unique is that rather than receiving rivers from upstream, all those things that appear to be rivers in Beaufort County are actually fingers of the ocean. Rising sea level has caused these areas to be flooded. And so these are tidal rivers because each of these rivers experience both 2 high tides and 2 low tides twice a day. They are very interested, since we're very interested in protecting the area that surrounds us, we're interested in the threats to the Port Royal Sound. And currently there are 3 that the prevailing paradigm is focusing on. One is citizen apathy. We need to create a culture that cares. 2 is oyster bed closures. That gets people's attention. Can we reverse that trend? And 3 is everybody talks about storm runoff. Is it too much, too fast? The question is that if we do all 3, is that enough to protect the Port Royal Sound? So let's look at these. First of all, create a culture that cares. This is one of the Spring on Trust big success stories. First of all, 20 years ago, we created the Port Royal Sound story. At that time, nobody recognized that our system was different from the other systems. And then after telling that story to Rotary clubs and Lion clubs and everybody that would listen for about 15 years, it became the story, the culture of the area. In the Master Naturalist program, here's a class of teachers in the Master Naturalist program. we were able to make sure that story was told to all the people who teach others. We were also able to educate elected officials. It was very gratifying to hear one county council member explaining it to the other council members when I happened to be present. And then the other thing we recognized is that this was something where the Spring Island community should not be supporting this effort when there are so many other communities that have resources. And so we were co-founder of the Port Royal Sound Foundation, which ensured that all communities were engaged with financially supporting and also with their volunteerism, this whole effort to create a culture that cares. The second area of concern is oyster bed closures. It's important to recognize that oyster bed closures are not done by the EPA, but by the Food and Drug Administration. And this basically occurs when fecal bacteria counts are too high, using a 3-year average. The assumption was that fecal bacteria are coming from human sources, but we now know they come from wildlife, they come from dogs. And so what does it mean when you have too many fecal coliform, according to the FDA standards, which are based upon edibility in the waterways? How does that affect Spring Island? Well, if you look at the red circle, that shows you where Spring Island is. And here are the oyster bed closures that are near us. And as you can see, on the other side of Kalawasi, that oyster bed's closed, and also the upper sections of the Okatee-Colleton River system have been closed. Just to zoom in a little bit, this little red line is where I put the road that leads from 170 across Kalawasi to Spring Island. So you have a better look at which areas are closed oyster beds. So some of the oyster beds close to Spring Island are those that are closed, which then causes us to be concerned. But it also raises this question: do restricted oyster beds indicate this area is unhealthy for marine life, or is it the fact that you just can't eat the oysters? Now, when we look and talk to people that are state agency people, what they tell me is often that closed oyster beds often appear healthier than open oyster beds because they aren't being trampled by people harvesting the oysters. So we know that healthy oyster beds, oyster reefs, indicate a healthy estuary. So is there a way that we can measure this? In essence, we're saying that bacteria in the water doesn't tell us about the health of the estuary per se, but the health of the community of the animals living in the oyster reef does tell us about the health of the waterway. There have been studies that have been done in Beaufort County. They found over 126 species of different kinds of invertebrates that live in oyster beds. It's an incredibly diverse group of organisms there. And so if all those organisms are healthy and you have high diversity, then you can assume that that part of the estuary is still healthy. Unfortunately, we have to destroy part of the oyster reef to inventory the animals living in it. And it's a very time-consuming process. So that's an issue, one that we're going to come back to at the end of our story. The third threat, we said, to the Port Royal Sound is stormwater runoff. Just to make sure everybody understands that when we say stormwater runoff, we're talking about rainwater that falls or collects on roads, parking lots, and roofs, and then flows through stormwater drains into a nearby tidal creek or tidal river. What happens when stormwater flows, when rain falls on a forested landscape? Then what we observe is that much of that water gets absorbed into the soil and therefore eventually ends up in groundwater. But a lot of that water also actually is taken up by the trees and vegetation and evaporates back into the atmosphere. So it doesn't go anywhere but straight back into the atmosphere. As a result of this, if you look at what happens on a forested watershed or landscape, only about 60% of the rainfall becomes runoff, and it's released over at least a period of about 36 hours. In contrast, when you have rain on a paved and developed landscape, notice that there's not the opportunity for nearly as much water to go into the groundwater, and there's far fewer trees and plants, so much less goes to the atmosphere. As a result, most of this water then ends up running off, so we officially call it stormwater runoff. The result is that 63% of the rainfall occurs as runoff, and what we need to notice here is it gets to the tidal creek or tidal marsh very quickly. So within 6 hours, we have this huge pulse of fresh water. Because it's flowing fast, that water is also picking up sediment, and sediment can have pesticides attached to it. So what are the solutions to stormwater runoff? Well, we can preserve natural wetlands, especially at the headwaters. We're very pleased to be one of the partners to help preserve an area called the Slater Tract, which is 3,800 acres right in the middle of the headwaters area at the top of the Port Royal Sound. They closed on that last week. We worked closely with Beaufort County to get stricter stormwater ordinances passed, some of the strictest in the country, about a decade ago. And the whole idea is to keep water on the land longer, which means now people are using stormwater ponds extensively, and the question then becomes, how healthy are they? Now, for us to look and see what the impacts of development are on the landscape, Dr. Eric Montie, who's a very productive and really impressive young scientist that came out of Woods Hole Oceanographic Institute, has done over the May River from funding that he received from the town of Bluffton. And this first graph actually shows how salinity in the May River goes up and down from year to year. And so this is one of the challenges, is that there are these cycles of El Nino and La Nina. So even a normal process, we expect salinity to jump up and down through the time period. The idea is that if you have stormwater flowing into a river, that it's going to cause the salinity to drop. He's using 30 years of data from DHEC that was taken in order to monitor the oyster beds. So that's the only long-term database that exists for Beaufort County to describe the environmental conditions here. And if you look at the bottom on the graph that is under A, that orange line is a lump of a lot of averages of fecal coliform samples taken by DHEC in the waterways. The one on the left is up in the headwaters that receives most of the storm water. And you can see over this 30-year period, there's very much a increase in fecal coliform. At the same time, the average salinity, the blue line, is decreasing. This is in contrast to the graph on the right, B, which was taken in the mouth of the May River, where there's a lot of buffering from the tides in the ocean as well. And you can see that there's been some up and down, but it appears that the pattern that we had 30 years ago still is holding for the most part in this time period, as has in the headwaters. On the right, what they did is they looked at land use, and pink areas show places that have impervious surface. So just look within the circle, you can see how development has changed the landscape up near where the graph made on left, near the headwaters, was taken in 2018. So development is certainly changing the type of stormwater runoff that's going into the May River. This is a paper that he has published in a prestigious journal, and so it's really top-notch science. There's another study that I want to refer to, and that is one that was done by NOAA a couple decades ago. And what they did was they looked at grass shrimp abundance and whether it decreased when exposed to pesticides. And that left-hand bar is the undeveloped forest. So when they looked at the grass shrimp that were present in tidal creeks receiving their water from undeveloped forest, that was the abundance they had. If you look all the way over to the right, if you look at my graph right here, you can see that when they looked at agricultural areas, the tidal creeks coming from agricultural areas had much lower densities of grass shrimp, and grass shrimp are very sensitive to insecticides. What's interesting is when we look at the rural single-family homes, Unfortunately, they were more similar to an agricultural area than they were to an undeveloped forest. And the lesson that we take away from this is lawn pesticides are one of the probably the number one threat to our tidal creeks, not golf courses. Think about the amount of land that is covered by lawns that are being managed to have nice turf grass and the amount of chemicals that are going into those areas. As Jay Grattan has described to me, they have much tighter restrictions working in these areas than people who are doing lawn care where more is better. Now then, so what fecal coliform paradigm, as I'm telling us, tells us that if we manage stormwater runoff, we will have a healthy estuary. The question is that enough? Is that true? The problem is we don't know how healthy estuaries work. As the saying goes, we don't know what we don't know. So with that, then let's go back. It caused me to go back and look through the literature, to reread things and also read a lot of new literature. And let's talk about what scientists do know about southern salt marshes. This work began in 1962, and I'm referring to it as the Spartina paradigm. The father of the Spartina paradigm is Eugene Odom, Dr. Eugene Odom at the University of Georgia. He's a legend. He died, I believe, in 2002, but he is known as the father of modern ecology. He wrote the first ecology textbook, and it is he who said the ecosystem is where everything is connected. He spent 60 years at the University of Georgia teaching grad students. And so most of the marine ecologists who look at salt marshes on the East Coast are either directly or indirectly connected to Dr. Odom. Early in his career, a second individual, John Teal, who came down and did a postdoc with Eugene Odom in the late 50s and early 60s, was also instrumental. He then spent his career at the Woods Hole Oceanographic Institute and was there from 1961 to 1995. So these two scientists really shaped the paradigms and the thought process of how scientists looked at the salt marsh. There is a classic book by John and Mildred Teal, published in 1970, called Life and Death of the Salt Marsh. And I believe I read that either as a senior in high school or as a freshman. That really got me first interested in salt marsh and their ecology. The main character in his book is the grass Spartina alterniflora. It's a perennial grass. Its roots send up new growth annually. These shoots die in the fall and eventually break off. These shoots become what we call floating rack. That represents a season's growth. And it's really important for us to think of that's the crop that it's produced every year. And rack is a form of detritus. And so the paradigm created by Odom and Teal proposes that the rack is carried by the tides out of the estuary and provides nutrients to the ocean. To appreciate the biology that goes behind how estuaries work, we need to learn a little bit more about it. There's a mass of floating rack that's going by the Port Royal boardwalk one day when I was out there. So let's look at this paradigm that these two scientists created. First of all, Eugene Odom said the salt marsh is an ecosystem, and that Spartina is the crop being produced. And one of the reasons they emphasize that is when you go into a Spartina marsh, there are very few herbivores. So there's some grasshoppers, there's a few snails, but basically the claim was that this annual crop was not being harvested by herbivores. Thus, you had a great deal of rack, this floating detritus, which is transported by the tides. And when they started looking at all the other organisms found in the estuary, they decided that they all had to be connected somehow to the crop because that was the source of energy. And so they started creating food webs. And the paradigm was stating that detritus is recycled by decomposers, provides energy and matter to other animals in the ecosystem. So all of us that have been trained as naturalists always talk about that Spartina is the source of energy for the salt marsh, and that the rack is how it's decided. I'm going to give you an alert. That ain't right anymore. All right, the paradigm has shifted. So to look at this paradigm, though, there are some things that are important. There's some background science info I want to give you. If you'll remember this, physicists say scientists measure only 4 things: matter, energy, space, by that I mean distance and area, and time. Humans can only observe matter and matter and space directly. They have to use instruments to measure energy and time. So we're going to be spending quite a bit of time talking about matter and energy. Those are abstract ideas to me when physicists start talking about it. So we need something more practical to look at. Food webs are going to describe the flow of matter and energy. What I want you to think about when we say matter is think about building materials, or like Lego blocks. In nature, atoms are the basic Lego blocks. And if you take atoms, which are represented by different kind of elements, they're different colors of Lego blocks. And in the real world, there's different kinds of atoms. And the ones that we refer to are carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. And so we talk about schnapps. When you hook these atoms together, you get a molecule. And so below, right here, we have a representation of a molecule where each of these letters represents a different atom. So all these atoms are connected together. Here's one molecule, here's a second molecule, and here's a third molecule. Now, in particular, when we're talking about nature and marine systems, particularly the salt marsh, there's one building block that is usually in short supply, and that's nitrogen. The pandemic has given us a good example of that. If you have a lot of material for building trucks, but you're missing one part, like a computer circuit on a chip, then you can't finish the truck. Similarly, in nature, you can have a lot of carbon and hydrogen and oxygen, but unless you have that building block called nitrogen, you cannot build ammonia. And once you have ammonia, ammonia is used to make amino acids, which are illustrated here. There's 3 different kinds of amino acids. Those are then hooked together to make proteins. So if you want to have living organisms, you have to have proteins, which means you have to have amino acids, which means you have to have a base molecule like ammonia, which means you have to have nitrogen. So that's a critical matter building block. In addition, you have to have energy. And energy, I want you to think of as the chemical bonds, which are actually little electrons zipping back and forth surrounding those two, holding the atoms together. And in particular, bonds between carbon and hydrogen are stored energy. So look at this molecule right here. Look at all those carbon-hydrogen bonds. That is a lot of stored energy. That's a fatty acid. Took 3 of those, hook them together, we get a fat molecule. No wonder it takes so much effort to burn a certain amount of fat. So we have matter building blocks, and we have energy, which are the chemical bonds that hold those atoms, that matter, together. It's hard to imagine, but the fact is that these carbon-hydrogen bonds are the energy of life when somebody talks about diet. The question is, what is that original source of this energy? Where does it come from? It's from the sun, as we well know. This is biology, but I'm going to give you a simple explanation about what plants do. Basically, plants get water molecules. They split water molecules. That's where they get the H's and the O's. In that process, they energize them so they can be attached to other things. They throw the oxygen away. That's a waste product. That's why plants give off oxygen. And then they use carbon from carbon dioxide, which is that gas in the atmosphere, and they hook all those carbons together to make big molecules. So we now have those being composed of carbon, hydrogen, and oxygen, where there are lots of carbon and hydrogen bonds. So when I show you a diagram like this, then what we're talking about is how do the animals at the top get their building material, their matter, and how do they get their energy? And we say it starts with solar energy, which are used by plants. Plants are eaten by herbivores. The little herbivores that are eaten by bigger animals. and those small animals then are eaten by the largest animals. So essentially what we have here is that it is moving up the food chain, so to speak. And so because it's moving from the bottom of the food chain up to the top, we often refer to this as bottom-up control. Now then, so what did Odom and Teal do with this? Well, Teal put a model together to measure the energy flow and connected all these together. And so one of the things about the Spartina paradigm is there's a lot of diagrams with a lot of kilocalorie joule measurements, but the whole thing is that they are all with the emphasis that detritus and Spartina is the source of this information. And in these models, there is a currency for the economy in terms of energy and nutrients, specifically nitrogen. So this is the unifying thing of the Spartina paradigm. If you want to understand how the salt marsh works, the idea is to map out where all the energy is coming from. So with that, one of the most important things to take away from this is you don't remember the details specifically, but an important lesson is that this Spartina paradigm saved the southern salt marshes. Because out of this, Odom had a message to the public. He was saying that science tells us this, that the Spartina marshes are the nursery areas for marine fishes and crustaceans. They're important to the seafood industry. And they use this information to have states create laws that fought to stop filling in salt marshes to create real estate and for digging them out for marinas. So that paradigm, that story, based upon the science, it was partially accurate and partially not, resulted in major decision making that occurred. So once Odom and Teal had done their part, and because of the role they said it played, then there was National Science Foundation, NOAA, became very interested in learning how salt marshes work. And so for the next 30 years, there was really the heyday of scientific research on salt marshes. And one of the questions that really they wanted to know about is the prediction that do salt marshes export nutrients in the form of rack to the ocean? So from 70s to 99, basically the research, the paradigm focused on how estuaries function. What's important to note here is that stopped by 2000. And since then, the focus has been on contaminants, sea level rise, and fisheries. There are basically three sources of information that I had to go to in order to look at how the salt marsh works. There's the research that comes out of the University of Georgia, most of which has been done at Sapelo Island. There is a 40-year study out of North Inlet, which was funded as a result of a marine lab created there by John and Winona Vernberg at the University of South Carolina. And the net result was there was a symposium held in 1999 called Concepts and Controversies in Tidal Marsh Ecology. So after this extra 30 years of research, what were the outcomes of the research? There are 12 things I want to share. Don't try to remember the details, but just look at how the story has been adapted and modified. First of all, the conclusion was that actually southern estuaries have three important crops: the phytoplankton, the microscopic algae that live in the water, benthic means bottom dwelling, the benthic microalgae, which are basically like what we see with the phytoplankton, but they're adapted for living on the bottom right here. And then the third is Spartina. One of the things that they determined was actually that, first of all, phytoplankton produces about 50% of the world's oxygen. So phytoplankton is a huge crop that's important for the ecology of the world as well as for estuaries. That the ones that live on the bottom, the benthic microalgae, which are also many of which are diatoms, represent more than 50% of the tissue eaten by small consumers. So rather than it being rack, it's actually these microalgae that occur. And third is that diatoms have an element in them called silicon, which also happens to be important in the structure of the Spartina. So silicon's another element to be of interest. The second thing that they learned is that Spartina's success of being able to live in such a hostile environment is based upon its relationship with soil bacteria. Spartina has nitrogen fixing bacteria like alfalfa and other plants, different kinds of bacteria, but they provide nitrogen to the Spartina, and in response, the bacteria absorb sugars from the Spartina. So it's a close working relationship. Also, there's a type of bacteria called Vibrio. It's one of the what we would consider a flesh-eating bacteria, but it's found naturally in estuaries. And when they get on the roots, they stimulate root growth by releasing a plant hormone, which makes for larger roots. And one of the challenges for Spartina is when growing in soil high in salt or hydrogen sulfide, that smell of the salt marsh that smells like rotten eggs, the toxic compound to living tissue. Spartina plants require extra energy to extract the ammonia, the nitrogen from the soil. This leaves less energy for growth. So what the bacteria do is very helpful with that challenge. So Spartina has a relationship with bacteria. Two, the marine fungi play a critical role in the estuary. And the reason being that fungi are a major decomposer. This just shows you the variety of different types of fungi that a team found just looking in one small area within the estuary. These are Petri dishes. Particularly fascinating is a study out of North Inlet where there's a red yeast, yeast being a type of fungus, that thrives in fiddler crab burrows. The fiddler crabs eat it. I guess think about portobello mushrooms are also a fungus. And their fecal pellets probably provide the yeast with nitrogen. So in essence, these fiddler crabs may essentially be farming red yeast living in their burrows. So it just illustrates the role of fungi for food. Now, when you see the rack, I now want you to think about an old tree that's rotting. And when you look at an old tree like the one shown here on the right, this is the area, this one has been dead for quite a while. See how porous this looks? That's because the fungi have digested the wood. When we say woody plants are comprised of cellulose, when I say that, think about celery, and then also lignin, which is wood. Bacteria and some protozoans can digest the cellulose and this hybrid molecule called lignocellulose, but it's only fungi that can decompose wood. That's the critical thing to remember. And so the animals can't do it. So the fungi are a key link in making it so that the fungi and that rack actually gets decomposed. We'll have to come back to the role of fungi later. The other thing that we want to look at is when we're looking at how the estuary works, is think about the Spartina marsh as functioning like a little miniature rainforest, providing this 3D surface area that provides all the surface area that's highly productive for all its inhabitants, which are mostly microbial. So all these stalks are coated with fungi. This dense miniature jungle, so to speak, provides hiding places for larval fish and juvenile fish. And on the floor of the estuary at low tide are all these little pools where the larval fish and crustaceans can safely spend low tide away from the big predators. And also that sediment, even though it's underneath the Spartina, still has algae growing on it. So there's a carpet of microscopic plant-like organisms covering it there. Now, these benthic microalgae, the diatoms that we told you that live on the mud, in essence create a pasture-like condition across the mud flats and sand flats of the estuary. And that material is then eaten by microscopic or very, very small animals. So these diatoms actually live in tubes. At low tide, they come to the surface so they can get sunlight. But then when tide comes in, they'll often go just under the surface of the mud so they don't all get eaten. Many of them are still eaten, and they divide either every couple days or sometimes they divide 3 times a day. The result is that this is a continually self-replenishing food source for those very small animals. What do those animals look like? Well, we call them myofauna. Myo means small but not microscopic. They're between a half and a millimeter long, And to keep in mind that what are they composed of? 70% of the myofauna are nematode worms, yuck, but they're microscopic almost, and these very small crustaceans that crawl around eating the algae. And then there's a mixture of other things. And when you go to a mud flat and you go to sand flat, you have different species of myofauna there. So we now created this image of all the mud flats and sand flats being like pastures for microscopic animals, and these animals that are like little grazers that are continually eating it. Now, one of the things that we're interested in is what happens when nutrients come in with stormwater. And some of the studies have shown that most nutrients are actually absorbed in the tidal creek marshes rather than being transported elsewhere in the estuary in solution. Remember that slide I showed you of how the main tidal river remained unchanged. And so what we believe now is that this high marsh absorbs this nitrogen-rich runoff. And other thing that's really happening is that it can either stimulate plant growth, so the plants take the nitrogen and put it into the soil rather than passing on, or when the soil becomes really high in organic nitrogen, the bacteria then basically transform it, convert it into atmospheric nitrogen, and so it leads the system. So this illustrates the fact that this high marsh, if we leave the vegetation there, is a great buffering system to protect the tidal river proper. The other thing that we're interested in is the fact those little tide pools in the upper marsh, if they get ammonia in those, that fertilizes those little benthic microalgae I told you about in the phytoplankton, which gives more food for their small consumers. So some nutrients within these tidal creeks and headwaters can actually be productive rather than harmful. The other thing we want to they've come to understand is that oyster reefs basically consume nutrients. They take things in rather than contributing nutrients to the tidal rivers. The oyster reefs consume zooplankton. That means they filter the zooplankton out of the water. But when they release nitrogen, apparently there's other things living within the community, all those little benthic algae and the algae covering that absorb that nitrogen before it gets into the main creek. The other thing is that in our systems, our tides are so high with so much volume that oysters do not play the role of improving water quality in southern estuaries like they do further north where they have much lower tides. The other point that we need to look at to understand how estuaries work is the fact that there are species, most of the species that we love from the estuary, shrimp, fish, crabs, are what we call marine transient species because they use different parts of the estuary different times. And one of the patterns is that they often will breed out here and spawn in the ocean, just off the, just outside the estuary during the winter. The larvae come in, mature to juveniles. The juveniles stay in the inlet until they grow larger. And then when they're large enough to be adults, they then migrate back out to the ocean. So the key thing here is, whereas Odom talked about the salt marsh being the ecosystem, the life cycles of these animals tell us that when we talk about it, it's really the estuary, which includes the near shore waters, that is the ecosystem. And when we talk about nutrients being transported from the salt marsh to the ocean, these studies that were done during that 30-year period showed that actually the nutrients are transported from the estuary to the ocean via all the schools of fish and crustaceans that grew up in the estuary rather than floating wrack. So when schools of fish move from the tidal creeks to deeper water and are eaten by larger fish, this represents nutrients being moved from the tidal marshes into the estuary and nearshore ocean. All right, numbers matter. Huge numbers of very small fish are getting eaten, and so they are all adapted to produce, females release millions of eggs. Well, these huge schools of fish and crustaceans are also releasing nitrogenous waste, a nice way of saying peeing, which has lots of nitrogen in it. And in the process, because we said nitrogen was a fertilizer, when they're in these shallow pools and all, they actually can be fertilizing the salt marsh and phytoplankton, the benthic algae in the salt marsh and the phytoplankton. So at low tide, there's a chance for that to be quite concentrated. And the key thing here is that if overfishing causes a loss of these massive schools of fish, then the microalgae may lose their source of nitrogen, of the nutrient cycling that goes there. One of the things that have been talked about is that during years where we have sufficient rain, they say we have a good shrimp crop. And the implication is that the nutrient-rich upland stormwater runoff coming through the groundwater may compensate for this loss of fish-derived nutrients in the recycling of tidal creeks. So runoff from the uplands, if in the right conditions and all, actually fertilize the process that drives the salt marsh. And then the last point is that seasonality is critical to the productivity of the estuary. You've heard me mention that certain things happen in the winter. Keep in mind that the winter's cold waters in the estuary cause few large fish to be present in the estuary. If the large fish aren't there, then that changes things. And then also, many of these species of marine fish spawn in the winter. The studies have shown that you have different phytoplankton communities are present in the summer and the winter. In the winter, the larger diatoms are the phytoplankton. And think about those are big enough that the little larval fish can see them and eat them. Whereas in the summer, you have very small photosynthesizing bacteria called cyanobacteria, which are the summer phytoplankton, which are eaten by protozoans. The protozoans are then eaten by rotifers, et cetera. And that we describe the fact that their standing crops of benthic algae and marine fungi are much larger in the winter. So that's what the research has shown. While all this was going on the East Coast, on the West Coast, there was an alternative approach to looking at food webs that is referred to as top-down control. This was originated from the work by Bob Payne at the University of Washington. He published this work in 1966. He's the one that came up with the key phrase, keystone species, to describe how zonation of the mussels, if you look at this diagram right here, this little band right here are mussels. And the reason is in this band right here is the upper limit is determined by the physical conditions that the mussels can tolerate. But this lower limit right here, he discovered was due to starfish who love to eat mussels, and they would line up here. And this was how high the starfish could go. So since this was the upper limit of the starfish, this was the upper limit of the mussels, he created this pattern. If you take away that top predator, then everything changes. And so over 10 years, he went out and kept removing starfish from these isolated rocks, and the mussel zone basically moved down. And so he also coined the term trophic cascades, meaning that the loss of a top predator causes dramatic change in ecosystem structure or nutrient cycling. And we'll come back to him in a minute. So are there any examples of top-down control on the East Coast? A guy named Pete Peterson, was doing a study in Pamlico Sound on scallops. I visited him in 1985 when I was just starting my career, and he took me out to these seagrass beds, and there were scallops all through the seagrass beds. And I thought that was so cool. 20 years later, I read a paper that he's written that things had changed. And that was the fact that in the 1990s, huge schools of cownose rays invaded Pamlico Sound and decimated the scallop populations, causing the local scallop industry to collapse. Why did all these rays come in? They have these crunching jaws. They love to eat shellfish of various kinds. When he started looking at it, he did some exclosures, found out if he protected sections of seagrass beds from cownose rays, the scallops came back and thrived. So it was indeed the predation. And then Peterson looked at what was going on at the same time, and then he realized that this coincided with the decimation of the region's large sharks. These large sharks love to eat cownose rays, and which of course are eating the scallops. So the loss of the sharks are the reason that there was a population explosion of the cownose rays and decimation of the scallops. A second example of top-down control was the salt marsh of 2010 to 2015. This was a combination of two things. One, it was during a drought period and therefore strained the Spartina, but also blue crabs disappeared. When the blue crabs disappeared, the salt marsh periwinkles became abundant. They made grooves in the Spartina and a fungus took over and killed the Spartina. This picture is from Shrimp Pond in 2015. So what's the state of knowledge today? There's a lot of information that focuses on causation in estuaries. There's no synthesis, but there was a call for a mechanistic approach to the study of estuaries. By that we mean, how do they work? So to do that, it's involved of coming up with a new story. And to do that, I've got to come back to Bob Payne. Bob Payne had grad students that worked on the West Coast studying the role of predators. And Bruce Mingy was one of his grad students. Once he got started his second year at Oregon State, he also got a bunch of grad students. One of them is this crazy guy down here. And so that's where I got my training, and that's my PhD. People ask me what I do. Is actually my training is in predator-prey systematics and community structure. So looking at this system as a community biologist, one of the things I realized is this lack of synthesis means they had the wrong paradigm. And so I asked the question, how can we look at estuaries in new ways? The answer was follow the fish. You needed to study their distributions, the different species and their abundances, because what the fish do reflect the nature of the estuary. And red drum is a perfect example. Here's a red drum that is adults breed in the fall out in the ocean. The larvae come into the estuary as juveniles. By December, they weigh a pound. They spend time in the estuary, and by the time they're 3 years old, they're large enough to go back and breed there. The other thing I looked at is the fact that they're dealing with a system that is structured in different organizational levels, and everybody kept treating different estuaries as replicates. That's not the case. So proposing a structure, I've talked to scientists, is that this region where the Spartina marsh exists is actually one population of red drum that move back and forth. So we treat this as what we call a biome. Within that, there's different individual estuaries that each are ecosystems. And within the estuaries, there's what we call economic zones. Within the zones, there are habitats. Then the habitats, they're localized networks. And within that are their symbiotic partnerships. Just like we talk about countries, states, counties, municipalities, neighborhoods. And let's take you through the cycle. One of the things that we need to look at here is the fact that in the winter, there's cold water, few consumers, we have lots of algae, diatoms, and fungi. And interestingly, we had peak phytoplankton crop right there when there's no predators there. The fish then arrive, the larvae and the fish grow fast during warm weather. In the fall, we have the peak fish and crab production. And so seasonality is really important. You can go out each season and see differences as well. Here's some winter pictures. We focus on these transient species, as I mentioned, and the model sets up the estuary into different zones based upon the raw materials that they produce, so to speak, which was illustrated by the red drum as well. In addition, with these areas, one of the things that happens in other estuaries is that the river has so much input on the water in the estuary that often magnifies things and makes it difficult to study these other interactions. That's why the Port Royal Sound system is so important, because it doesn't have that variable. And each of these zones has a place where prey can escape that I've described to you. And in addition, each zone has top-down predators that influence the abundance of those mid-level consumers. With the exception of oyster reefs, they actually have a sponge that digests the shells. That's why we don't have any oyster shells in the area below there. We then have to divide the estuary into different habitats and have both a summer and winter approach to it. And from all this, we end up with a new list of vulnerabilities based on this new paradigm. One that comes out is actually overfishing of fish and crustaceans could have dramatic effects on the whole system because of the loss of estuary nutrient transport and also these top-down predators. In addition, stormwater, if it channelizes and washes out these tidal creeks, you lose the refuge for the small ones. there are chemicals that we apply to golf courses and lawns, like fungicides, nematicides, insecticides, that could all kill key players in the food webs, now that we know the roles of fungi and nematodes, as well as our crustaceans. And another one that's of concern is that there are hormone disruptors in tidal creeks that can affect fish development. Of course, river pollution is always a problem in some places. So with that, where are we going with future research? The things that the Trust has been supporting that help elucidate how estuaries function is a dolphin survey with underwater soundscapes, an analysis of the DNR has a great database for long-term fish surveys. They can't do any work with that because it's not under their grant, but if provided funding, they put a grad student on it, and they also do an analysis of the reef communities. And then finally, the study I told you about where Eric Montie looked at the May River is that he was able to get a grad student, and they are now in the process of doing that for all the tidal rivers in Beaufort County. And that's going to be a very important tool for us to be able to go to county council to sway their efforts in terms of helping them recognize that they've got to do something to manage development and the restrictions that they put on those. So with that, I'm going to stop sharing my screen. It was a lot in a short amount of time, I recognize, but it's a story that has lots of parts that you needed to get an overview in order to understand the importance of trying to figure out how they're all connected. So with that, leave a few minutes for questions. Sorry, it took a while.
So first of all, can you tell us how dolphins help us tell health of the creeks in the estuary?
Yes, dolphins have the same physiology as humans do because they're mammals, and so they're sensitive to contaminants. In areas that are polluted, a female's first baby usually dies because all the pollutants they're fat soluble, they tend to go into the embryo, and that just illustrates how dolphins are sensitive indicators. Our dolphin surveys here are looking at survivorship of the young, and so we don't think that happens here, but the study's still early.
Okay. You talked about the drought period from 2010 to 2015 and marsh dieback. What brought the marshes back after that? Was it a function of the drought? Did that?
Yes, it turned out that after the drought was over and we started getting regular rains, then one of the things we saw was that the Spartina plants weren't as stressed. They started recovering some. But also we saw a dramatic return of the blue crabs. It was very difficult to catch blue crabs in 2015, and now they're common. And those areas, when you come across the Spring Island Bridge from Kalawasi and you look to the left, that's an area that was part of the die-off that's recovered.
Yeah, I think it's worth mentioning. Hopefully my sound is not causing any trouble. I got a little feedback.
Oh, that's good.
Okay, but I think it's probably worth mentioning that there is a good association between blue crabs and drought as well. They tend to like to breed in the slightly less saline areas. So when you get drought, you have problems with blue crab reproduction.
Right. Exactly, yes.
Okay, so another question is, you talked about the effects of fertilizers and nutrients going into the marsh. If you use organic fertilizers, does that change the effect?
It increases the rate of uptake, which is important. The key thing with using fertilizers is not to use it before it rains and not to put too much out. Fertilizers are not the problem, it's excess fertilizers. So only use as much as your plants can absorb, and so the organic fertilizers, I think, are more efficient with uptake than the inorganic ones. That's a good question for Carl to confirm that.
Yeah, and that may be the problem with inorganic fertilizers, maybe the production method as well. I think that's more harmful to the environment than an organic, but the ultimate effect on the marsh may be a wash because it's nutrients or nutrients.
Right. The whole issue is depending upon some low dosages, getting to the marsh might actually be beneficial. But when you get too much there, you shift from having these healthy diatom populations to a lot of filamentous type cyanobacteria.
Okay, so we'll do two more questions. One is you talked about the fecal coliform bacteria in oysters, and you said if there's bacteria, does that mean that the oysters, is there a contaminant problem, or is it just that you can't eat the oysters? And do you want to elaborate a little bit on some of the initial information we found out from Alan Warren about the health of the marsh and a different type of testing that you can do?
One of the things that they're looking at is that there's different type of bacteria, and we're going to use those as a tracer. One of the things you can do is you can use bacteria as a tracer to find out where you're getting stormwater. The oysters themselves appear to be healthy, so it's not the bacteria that's creating the problem for the oysters, but the question is, are these other chemicals coming along with the stormwater that's affecting the causing the fecal coliform to come up because there's more fertilizer in the water.
Mm-hmm. Okay, so last question, what we'll conclude with is what can individuals do to help with salt marsh health, given all of this stuff that you just talked about?
One of the most important things that we do is, and in Spring On is good about this, is we try to minimize the amount of lawn. You know, lawn is a very beneficial thing to have if you're a homeowner in terms of having a place to walk if you want to, but making sure that you don't have lawn near the edge of the marsh and that you maintain a healthy buffer between any lawn that you have in the edge of the marsh. That allows a chance for these plants to absorb the chemicals that can be detrimental. The second is that if people are concerned about insects, sometimes people put up these misting systems. The material that comes out of those misting systems is highly toxic to larval crabs and shrimp. And so those should never be close to the marsh edge. So keeping those things away from the marsh is the most helpful thing that individuals can do.
All right. Well, thank you, Chris, so much for, I guess this is our last Trust Talk of the spring season. We're planning to do a talk once a month through the summer, so do stay tuned, but this is the conclusion of our spring, and so I'll let you close for the evening.
Okay, and I just want to again remind everyone that the Trust Talk Committee heavily relies on suggestions from Spring Island members for Trust Talks. It's particularly helpful when somebody knows somebody and is willing to host them. We do plan to go back to in-person Trust Talks starting in September. We may continue to have some that might be a Zoom talk, but it'll be one where everybody can come to the summer house and sit and watch it together if we get you know, an exceptional speaker who's somewhere else in the world that we cannot bring to Spring Island. So with that, everybody have a great evening, and we'll give you a heads up when the summer Trust Talks are going to occur. Thank you for your patience, and I hope it was something that you found informative and entertaining.
All right.
All right.